everybody, welcome to our webinar, Stinkier Situation, Controlling Odors in the Cannabis Industry. If you joined us a couple years ago, we had part one of this webinar, A Stinky Situation, with Caitlin Urso. And so we wanted to update everyone on where we are, where the research is and the technology is associated with controlling VOCs from cannabis plants. So just a real quick bio of Dr. Greenhouse. I think most of you know who we are that's on this call, but we're an agricultural mechanical engineering firm that designs HVAC systems for indoor farms and greenhouses. We've been in business for a little more than 5 years and have done almost 150 projects across the US, Canada, and a few countries. We've been on every continent except Australia and Antarctica so far. So if you know of any projects out there, we want to go there. For those of you who may not have heard, we have our next in-person HVAC workshop, May 5th through 6th here in Sacramento. Dr. Alex Guenther is actually going to be one of our guest speakers, talking about odor control and the quantification of odors. It has morphed into a fully 2-day event with lots of group activities and a whole lot of teaching by yours truly. So if you want to learn more about that and you think you want to come, it'd be great to have you. All right, so without further ado, let's get started. So we have special guests. I get to sit back and listen and moderate, which is very cool for me, and hear from these experts in the industry. We're very focused on odor mitigation and quantification in our industry. Our first guest speaker is going to be Dr. Alex Guenther, who is a senior scientist for Byers Scientific. He has a long storied career in quantification and measuring, what biogenic emissions, as well as their impact on the Earth system. He worked for NCAR, the National Center for Atmospheric Research, for more than 20 or 30 years. I guess that's 20 years, and is now a professor at UC Irvine and is working really closely with Byers Scientific to quantify these VOCs that are not very well studied yet. So I'm really excited to hear about his updates for all of us on where that stands. And then we have Emily Long, who's a senior project manager with Byers Scientific. And she's going to be talking to us about targeted mitigation of these VOCs. She's really active in NCIA on the Scientific Advisory Committee, has also been working with the Cannabis Sustainability Working Group to develop best management practices for the industry. So we have some really great people here on this panel. I'm really excited to learn from them. And without further ado, I'm going to hand it over to you, Emily. So I'm going to stop sharing.
All right. Thank you, Nadia. I'm going to start sharing. So just bear with me 1 second.
And
you guys,
as she's setting up, sorry, just real quickly, if you have questions, please use the chat feature, and we'll follow up at the end. We'll definitely leave some time for Q&A at the end. And it's up to Emily and Alex if they want to field your questions in real time or not, but let's use the chat for now. And we can always unmute you also if you want to ask your question with your voice. All right.
Awesome. Thank you so much, Nadia. First of all, I want to thank Nadia, Dr. Greenhouse, for including us in your webinar series. We're excited to update you on the state of the science when it comes to cannabis VOCs and odor emissions, as well as the industry-leading control technologies, what we believe to be so. And of course, Dr. Guenther, it's always a pleasure to present with you and work with you. So I look forward to this webinar. I don't have a slide for this, but just to lay out some context because the audience, you guys may see references to Byers emissions analysis, Byers scientific. So just to lay out the landscape for you, Byers Scientific has 3 divisions: our Byers emissions analysis, our mitigation technologies, and our intelligent systems. The emissions analysis side, which is the side that Dr. Alex Guenther is our senior scientist of and our lead team member, we research and study the cannabis plant emissions. And then that data and that research allows us to engineer and design our control technologies that are best suited for cannabis emissions control specifically. And then our intelligent systems division is just allowing that mitigation technology to be intelligent, which allows for more energy efficiency and less waste. And also real-time monitoring and reporting for compliance purposes. So that's just kind of, if you see different Byers names there, that's just the explanation of kind of how we all work in synergy together to help the industry. And now I'll just quickly go through this overview, but this is what was in the promotional material for the webinar. So this is really just speaking to what we're going to cover today. So obviously, what are these VOCs? Why are they produced by cannabis and by a lot of plants, all the plants? Why is it important to quantify these? And how are they quantified? And so Dr. Guenther will go into all of that, as well as the characteristic skunk-like smell of cannabis and what that exact molecule is, as well as we'll speak to some of the current standard development and regulations or lack thereof around odor control. And then I will then speak at the end of towards the end of the presentation about how we can effectively reduce odor complaints with proper custom designed mitigation controls. And then we'll talk about how we can do that efficiently and sustainably, reducing energy use and carbon waste associated with a lot of the practices with odor control currently in the industry. So without further ado, our senior scientist, Dr. Alex Guenther, is a world-renowned air chemist who has studied plant volatiles for over 30 years and is now bringing his wealth of knowledge to our wonderful cannabis plant. So we're very appreciative to have this level of research and understanding. So Dr. Guenther.
Hey, thank you, Emily. And as Emily told you, we're going to start with some background on plant volatiles, these chemicals that plants emit into the atmosphere. And with many of these compounds, we understand them pretty well. We know where in the plant they're produced. We know the biochemical pathways, how plants are making them. We understand the physiological mechanisms that control their emission. But for some, we don't know them very well. Some plants have been well studied and others not at all. And yeah, and go ahead, next slide, Emily. And as far as why plants emit these, well, some are real obvious. You all know about the birds and the bees, and of course, floral scents are attracting pollinators. Fruits often have odors for attract seed disseminators. Some of these compounds are for physiological reasons, hormones, for example. Some are protection from insects and herbivores. Others just from environmental conditions. For example, some plants have sunscreen. They produce sunscreens, some of the same compounds in our commercial sunscreens. So a lot of different reasons, but again, with some compounds and some of the main emissions, we don't really understand very well why these compounds are being produced and emitted in large quantities into the atmosphere. So next slide. For myself as an earth system scientist, I've been very interested in what the impact of the, what's the impact of these compounds on the environment. And in a lot of cases, in a clean environment, these compounds are emitted into the atmosphere. They get chewed up by oxidants in the atmosphere and down to carbon dioxide, which is then taken up by the plant again. Next slide. But interestingly, in a polluted environment, you get something very different. In a polluted environment, these plant chemical volatiles will enhance the formation of pollution, of smog, ozone, and particles. That's something we've studied in the US and Europe. But a real interesting place that we've had to study this has been in the middle of the Amazon. So in the city of Manaus, Brazil, about 2 million people, very polluted. We've set up sites with aircraft and field sites around there, and where we could, when the pollution from Manaus is coming over the field sites, then we can see what happens in a polluted environment. When the wind blows the other way, we can see what happens in a clean environment. And so we can understand this, that how these plant volatiles do very little in a clean environment, but they do a lot. They have a very big impact in a polluted environment. Next slide. And the amount of this emission is tremendous. It's in gigatons of emission. And I'm showing here for the global annual total, you can see the emission of these what are called volatile organic compounds, VOCs, is 8 to 9 times more from vegetation than from all the car tailpipes, smokestacks, power plants, solvents from industry. So on a global scale, it's much greater. So this is why for climate models, for large-scale air pollution models, we've been very interested in these emissions for a long time. But we've always considered the emission from urban areas to be relatively small. And 30 years ago, that was actually true. But in the last 30 years, we've been greening our cities, which is a very good thing. But also we've been cleaning up the air there, which again, a very good thing. But now we're at the point, if you look at the top pie there, where the emission of these VOCs from vegetation in Los Angeles County, so about 10 million people, one of the most populous counties in the US, is about the same from vegetation as it is from all the cars and all the other anthropogenic emissions. But if you look at the bottom pie there, that's looking at the ozone or smog formation potential. And so all these compounds are not the same. The ones coming from the plants actually have a greater ozone formation potential than the anthropogenic emissions. So looking at that, they actually contribute more to smog. But again, going back to what I first said, and if it were a clean environment, they would have very little impact. So it's not the fault of the plants, and the solution is not to get rid of the plants. If we can clean up the air, the oxides of nitrogen and sulfur and other things, then these plant volatile emissions will not have any impact. But as long as we have polluted air, then it's a real problem that we work hard on trying to understand. Next slide. And we do this by creating quantitative models that go into all the global climate models, and the regional air quality models use these. The ones that I've been working on developing is called MEGAN, the model of emissions of gases and aerosols from nature. And so we have this so we can predict anywhere in the world at any time, including predicting future emissions, predicting past. And in this model, we need to know some things about the landscape, what kinds of vegetation you have there and how much. And then we need to know various environmental parameters that control how emissions change with temperature and light and CO2 concentration and stresses, for example. And next slide. And so we developed these numerical models through observations, of course, and this includes on very small scales where we'll do from looking at individual cells or in leaves. We look at the whole ecosystem and in canopy scale studies, fly aircraft around, for example, in the study I mentioned in the Amazon, and then up to the global scale where we rely on satellites measuring chemical concentrations throughout the world and on a daily basis. Next slide. But a lot of our fundamental understanding then comes from the very small scales. And so this enclosure technique that we've developed, where we can take individual leaves of a plant, or in some cases a whole plant, and put it in an enclosure, control the environment, and then study exactly what that plant is doing and how it responds to light and temperature and other environmental parameters. Next slide. And from this, then we developed these semi-mechanistic algorithms that do a pretty good job of getting the main controlling factors. The next slide. But then there's cannabis, and cannabis is something. So I've worked with actually on all 7 continents, done field research and made measurements, thousands of different plants. Cannabis is really unique. It has a greater diversity of chemical compounds and greater abundance than really any other plant that I've ever seen. And of course, one of the first questions that many people have is, what is that odor? What's causing it? It was really interesting to me that just a few years ago, nobody really knew. If you asked people in the industry, they often thought it was the terpenes. Myrcene is often a dominant terpene, and some people thought it was that, or one of the other dominant ones like alpha-pinene or limonene. But when we at Byers and we came and looked at this and we thought, well, it's clear no terpene has this odor, and it seemed unlikely that a combination of terpenes could come up with that kind of odor. It was far more likely that it would be something like what causes the odor of a skunky beer or even skunks. Next slide. And that is our thiols, organic sulfur compounds. And so we thought it was likely to be that, but it was challenging. Now for myself and working in federally funded labs and in academia, we can't study cannabis. I can't have cannabis on campus or in my lab and look at it. And that, of course, is a big part of the reason why people really hadn't gone and figured out what that odor was. But with the opportunity and working with Byers, we were able to nail that down. Next slide. And by using state-of-the-art analytical techniques, we were able to do a full analysis of all the compounds emitted by cannabis. We look at several hundred different compounds. Although looking for these thiols in the cannabis was really like looking for a needle in a haystack, because these thiols are emitted at 1,000-fold and even 1,000,000-fold lower concentrations than these terpenes. So it makes it really difficult to get this through an analytical system and look at it. But we were able to look at sort of the full suite of thiols and also some of the oxygenated compounds and nitrogen compounds, which also can have odors. We looked at a variety of these and determined that this 321 MBT, it's also known as prenal mercaptan, seemed to be a dominant one that we would see in the skunky odor cannabis. And it made sense because in the authentic standards that we had, it smelled very much like that skunky odor. Next slide. But by using these authentic standards and comparing it to NIST databases, we were able to determine very precisely that this is the compound coming from cannabis. Next slide. We took this a little further though, and collaborating with our colleagues Don Wright and Jacek Kozil, and so Byers sponsored an effort to look at including hemp odor, and we're able to determine using both our state-of-the-art analytical techniques and their approach, which uses the human nose. So this is called GCMS olfactometry. And as you can see over on the right there, that's the operator of this system, and they've got a hose hooked up to their nose. And as the compounds elute from the GC, they describe the intensity and what kind of odor. And so there was just one that came out as a skunky odor, and that matched up on our GC as being the one that we could definitely identify as the 321 MBT. Next slide. And one thing I wanted to point out here, looking at these two traces here, there's a red trace, which is the standard mass spectrometry that's used. And the top one is the black trace. That's that one coming from the human nose. And if you look over on the right there, where there are these big peaks 11, 12, and 13, that where the human nose saw these really strong distinctive odors, the standard mass spectrometer saw nothing. Now, using our advanced mass spectrometry, we can detect there, but standard techniques wouldn't even see anything there, which explains why other people who had looked at this weren't seeing anything. Next slide. So with that success, we've actually moved forward and contributing to an effort led by our colleagues, Jacek Kozil and Don Wright, in developing a standard approach. So ASTM is an international organization that develops standard approaches for things like environmental odor assessment. And so this is under development, but it's a combination of the tools that they use, like the olfactometry, the human nose, and the state-of-the-art analytical tools that we use, like time of flight mass spectrometry. Next slide. Another approach which they've pioneered that's an important part of this is called the rolling unmasking effect. And you may have noticed this, those working in the cannabis industry, that sometimes right outside the facility, you don't have such a strong sense of that skunky odor, but you go downwind a block or two, and then it comes out really strongly. And what's going on there is that essentially the odor is masked by these other compounds, such as the terpenes. And then as you go downwind, you have a different ratio there, and then your nose perceives this very, very strongly. And so this recognizing that these different ratios and different amounts are really important as well too, that, and you have to bring in a nose to look at that. Just the most state-of-the-art instrumentation couldn't tell you this. Next slide. And so that's really the key in this approach, that we bring together the state-of-the-art instruments and the human sensory nose as part of this. Next slide. So kind of summarize that, there's different plant volatiles will have a very different characteristic odor. Of course, alpha pinene, the piney odor from a pine tree, alpha humulene, the spicy hoppy odor in the beer. But a lot of odors are a mixture of compounds and can be very complex mixtures. Next slide. And I'm sure those of you in the industry know this very well with cannabis, that there are very different odors. And of course, clients are very interested in having these different odors. So we recognize it's very important to the product. But it also means that the emission rates from different strains are very different, both the magnitude of the emission, but also the relative amounts. And so just one measurement from one cannabis strain isn't going to really tell us everything that we need to quantify the emissions and understand where the problem might be outside the facility or a block downwind. And so in our assessment, we assess these strains and we look at these 12 different categories, each one of which may have 5 to 50 different compounds in them. Now, most of the mass here are terpenes, but there are over 200 compounds, and many of them contributing to the odor are these very minor compounds like the thiols. Next slide. And so what we do, we've developed an approach we call the canopy emission profiler approach, and using those enclosure techniques that I mentioned, and using the models that are based on past 30 years of working with these plant volatile emissions and developing numerical algorithms that we put into climate models and air quality models. We've taken that approach and applied it to cannabis. So based on our understanding and studies of what controls variations, then getting the amount, the density of the cannabis in a facility, and then, of course, importantly, what type of strain it is, with the recognition that each kind of strain can be different. And next slide, and I think now turning it over to Emily.
Yes. Thank you so much, Dr. Guenther, for all that wonderful plant VOC knowledge brought to the cannabis industry to help advance and move the industry forward. Thank you so much. I will kind of jump into taking off where Dr. Guenther led off, discussing the reason for the quantification of these emissions. Why that is so a lot of the current regulatory frameworks are built around uncertainties. Odor is very subjective, as we all know. So in order to make sound policies, we have to base them in science. And these emissions profiles, as Dr. Guenther just said, they vary by plant, by species, by the stage of development of the plant. There's so many variances. So this bottom-up approach is so important and key to quantifying these emissions, and then therefore using that cannabis emissions profile that Dr. Guenther just spoke of. That's a facility assessment. We're able to calculate not only the emissions coming off of the plants, the canopy, but the actual entire facility. So that entire emission load is then able to be calculated. So that bottom-up approach is very critical to support the industry and allow policymakers to create sound regulations that are based in science and not for example, a nasal ranger, which is highly subjective at identifying odor in cannabis. And it's interesting, actually, just anecdotally, we come across this all the time in the field. There are so many regulatory bodies in different states where both recreational and medical cannabis are legal, that they are going through these license processes, these hearings, and they don't mention odor. They don't bring it up all the time. So it depends, but there's a lot of times there are these massive issues in the media of these large MSOs having, many people frustrated about the smell coming from their facilities, but yet when an application is submitted, there's just no mention of it with the regulatory bodies. And it just happened again today at a hearing we were involved in, or yesterday, excuse me, where there was no mention of odor, but the community was there representing those stakeholders, right? And they were like, what are you going to do for odor control? And the actual policymakers, the planners for the city weren't talking about it. And I bring this up just to say that by ignoring the issue and pushing it off just because they may not understand that there is a solution, like we're explaining, it's just going to mean that the federal government will step in eventually and create a lot more red tape for the industry. So this is overall not a good move. And that's part of what Byers does, is we'd actually do consult with a lot of these regulatory bodies in these various states as they come online and legalize cannabis, to try to help them understand this bottom-up approach to emissions quantification. So I don't know if you really went into the leaf enclosure method, but this leaf enclosure method was actually perfected for cannabis applications by Dr. Guenther himself. And so we actually go, when we do these facility specific measurements to reduce these uncertainties, we go in there, we actually enclose part of the plant, purge all of the other air so that we are only measuring what is coming off of the plant, what the plant is breathing, using these measurements. And then we're able to calculate emission rates based on those measurements that we take of the various strains and the various life cycles at those facilities. I want to talk a little bit about carbon. I think it's a very misunderstood, and what we see this, it's a very misunderstood and very important aspect of VOC and emission and odor control for a lot of industries, but certainly for cannabis. And I want to talk about it with regards to efficacy and also environmental sustainability from a sustainability standpoint. I'm sure some of you are aware, but maybe not others, that the carbon is actually, the process is called adsorption for capturing these molecules, these emissions. So add with a D, and that's a process known as van der Waals forces. And so the actual physical properties of the carbon itself is very important. Mesh size and porosity is incredibly important. The carbon that Byers uses in our systems. For example, a single tablespoon is the equivalent surface area of an entire football field. And so that we know is going to be effective for having the proper surface area for the concentration levels that we're seeing at these, to be able to absorb those emissions and at those concentrations that we see. Another note, and this is kind of more on the sustainability side, is the carbon housing. So a lot of times with can fans, for example, which is widely used, I know Nadia, Dr. Greenhouse, and I were laughing about this on our original call. They're used way too often, and there's an arbitrary number of them. They're placed all around the greenhouses, and they're using plastic. They're made of plastic, so they're going to end up in a landfill. We use metal cylinders, for example, that can be refilled. And one of the issues of, we work in other industries, we work in solid waste as well, Byers Scientific. And one of the issues of these can fans and the spent carbon, especially from manufacturing facilities where they're using large amounts of solvents for extraction and post-processing, that spent carbon will actually get into, it's not an emissions issue necessarily at the landfill, it's the going into the water. So the leachate, which is the wastewater around these landfills, will actually be contaminated with that spent carbon and those solvent VOCs from that carbon, and not to mention the plastic. So one of the new projects that Byers is working on is actually, we're able to refill and recycle these cylinders. So the cylinders that we use, the housing is metal, it's reusable. And we're able to, obviously cut costs, but more importantly, diverge a lot of waste, carbon waste from landfills. So going back to the importance of carbon, we have residence time. So obviously how long the emissions, the compounds are able to, how long it takes them to pass through that carbon is going to be very important to how effective the carbon scrubbing is. The activated coconut shell is what we use specifically in our systems, and that is specifically carbon designed to absorb the concentrations of emissions that we're seeing. A lot of times you will see bituminous coal. That is what can fans have. That's a lot of times what we see in the industry. And unfortunately, that is not as effective at all. Just with surface area, it's not the, doesn't have the power of the coconut shell, the activated coconut shell carbon that we specifically use for cannabis emissions control. And another thing that I want to mention is, when it comes to carbon selecting, excuse me, buyers beware is kind of a silly term, but we do recommend that you ask for third-party test results of the carbon in a cannabis application, because of the uniqueness that Dr. Alex Guenther just spoke to about these levels of VOCs coming off of these very unique and complex plants. That setting with those high humidity levels is not the same setting as where carbon may be applicable other places. So bituminous coal may work in another industry, and it does. However, if you ask for a third-party test of your carbon supplier, if you will, in a cannabis setting, we recommend that you do that. You may not like what you find, but that is very important. Another aspect is pre-filters. So, in our molecular filtration systems, for example, we have a pre-filter before the carbon set stage, the carbon scrubbing, and that pre-filter captures the moisture in the air, which is, as you know, in these high humidity greenhouses, that's very important. But those moisture molecules will actually take up seats on the carbon surface area. So if you think of it as like a stadium seating, it's going to actually take up those seats, and then the carbon won't be able to absorb the odor. It won't have as long of a life because all those seats will be taken up by the humidity. So that pre-filter really acts as it allows the carbon to have an extended life, which again reduces the carbon waste that would come out of that system. And then lastly, we do butane life testing of carbon, which is so important for sustainability and to reduce the carbon waste again. And what this butane life test is we actually will send a cylinder off at, we usually do it at 6 and 9 months with our clients. at those 2 different periods of time, and we'll send a cylinder off, and they'll do a butane life test, which actually shows the remaining life of the carbon. And with that information, you're able to dial in exactly when you need to change out your carbon, that carbon replacement, which will vary by facility size. Obviously, it's going to vary by, your canopy size and how many, what the concentration levels are, what the humidity levels are. So understanding that specifically at your facility is so important to eliminate breakthrough, for one, so that there's no community, complaints due to lack of changing out your carbon. So for all of those reasons, really just understanding carbon for the industry is so important. I know Dr. Guenther talked about this, but this is really the summation of why we do these facility assessments and this bottom-up approach to emissions quantification. This cannabis emissions profiler, which was developed again by Dr. Guenther, is this mass balance calculation specific for cannabis operations. And we're able to use the volumetric measurements of the space, the emission load coming from the facility, and we are able to right size or dial in the mitigation technology using that data, which saves energy cost, reduces waste, and will actually be more effective at removing VOCs and odor. So this is just an example of the application of the CEP. So for example, a 225,000 cubic feet cultivation space, with put into that calculation all of the volumetric measurements, the emission load, it indicates a minimum of 3 air exchanges per hour, which allows us to then figure out, okay, we need 2 6,000 CFM molecular filtration systems, or 12,000 CFM is required to scrub the air at the rate at which it's emitting at that specific facility. And so that level of dialing in our systems is unique to us. And thanks to the CEP by Dr. Guenther. Our MT series of molecular filtration systems is one of the leading technologies in the industry right now. We have the ability to do anything from 2,000 to 6,000 CFM, depending on obviously the size of the facility. So we have these various sizes of molecular filtration systems. We have our proprietary pre-filters, which I mentioned earlier, and how important that is for extending the life of the carbon, capturing that moisture and those larger particles so that you can save the absorption qualities of the carbon, and as well as improving indoor air quality. We actually, in the 6,000 CFM unit, which is one of our larger units, that's actually a picture of it right there. We have 48 of the 24-inch coconut shell activated, the metal canisters. And again, talking about the surface area and the absorption quality to remove the optimal number of VOCs talks to the importance of carbon. Another unique and recent offering actually is we recently partnered with VFA Solutions based out of the Netherlands. It's Dutch technology, this Aspra. It's a very unique approach to ionization, to electrostatic, it's called electrostatic precipitation and filtration. And I'll get into that in more detail at some slides coming up. But within our molecular filtration systems, we have the ability to incorporate a phase using Aspra, which allows us to really focus on indoor air quality and plant health, and not just the carbon scrubbing, not just the odor. So now we're looking at complete package for a very comprehensive mitigation technology for all of the emissions that you will see in a greenhouse setting, and indoor as well. So this optional unit, which doesn't require, or optional phase, if you will, which does not require, we're able to manufacture that at our facility and put that in for our clients. It will allow for indoor air quality and odor control in one system, which is more energy efficient than having multiple units running on the electric grid and just sucking up energy costs. Another quick mention here is that the only requirement for adding in Aspra is replacement of the filter, which is once a year. these aspirate. It's very minimal maintenance and also very energy efficient. So the system overall is extremely energy efficient. So we have our third-party testing and validation of our molecular filtration systems. Specifically, the MT6 was what was looked at. I'm sure a lot of you guys are familiar with SES engineers. That's who did the third-party test. We looked at 10 days of measurement in a commercial cannabis cultivation. We had an expert odor panel participate and do the evaluation as well. And there was a no determined, or the 321 MBT that Dr. Guenther spoke about, which is the culprit of the skunky smell of cannabis, was reduced by 95% and determined undetectable downwind from this third-party testing and validation. So the system works. I want to go into a little bit more detail here with Aspera. We are really excited about this technology for the industry overall. Not only from a sustainability standpoint, having a comprehensive system really focused on plant health, air quality, and odor, but really clean air is a right. And that's ironically the president of VFA. That's her. She said that recently on a podcast, that clean air is a right. And that is such an impactful statement and so true. And I think a lot of people are now thinking about that more and more, especially because of the recent pandemic that we all went through. I think people are really concerned about, what is happening in the air around them and what are they breathing. So indoor air quality is so important. And I understand that there's a lot of fresh air coming into a greenhouse, but I'll get into that in a little bit. So in a greenhouse application, the removal rate is inversely correlated with the amount of fresh air. So meaning during blackout, that's when we would run a molecular filtration system with Aspera included, with Aspera inline. So we would actually run it during blackout, and then the efficacy rate would increase of the system. Once the ridge vents are opened, the efficacy rate would slightly decrease, which is why we typically recommend ridge vent vapor applications as kind of an insurance policy for odor with greenhouses. We'll actually install our proprietary waterless vapor system along the ridge vents so that we can capture anything that when you burp the greenhouse, there's not going to be any odor complaints. But point being that we really would run these systems during blackout, and that's when their efficacy would be the highest rate. So Aspera technology, it's closed ionization. I think that is a really important concept to kind of go into a little bit. It's controlling VOCs in a single energy efficient system. And with open ionization, you have the chance of byproducts to be created, right? But when it's closed ionization, which is what electrostatic precipitation and filtration is, what this technology is, it's all happening within the cylinder, which you can see right there in the bottom right-hand corner. So the particulate matter is captured inside of that system. It doesn't leave that system. So it is really a safe chemical-free alternative for protecting your plant health. It doesn't stop the creation of powdery mildew and botrytis, which is important to talk about, but it does stop the spread of it, which we know is a major issue in California and a lot of other greenhouses in other states as well. Move on here to, actually, no, I want to go back really quick and just mention when these different particles are going into this electrostatic field, which is kind of what you see here with these yellow whiskers, if you can kind of make out, I don't know if you can see my cursor, but in that bottom right image, whether the particles are negatively charged or positively charged going into the system, or whether they are dead or alive, a live virus or a dead piece of dust entering the system, they are charged positively and therefore permanently deformed. Their molecular structure is completely deformed permanently. And they then are going to be either, depending on their outcome charge, they're either going to be attracted to the surface that metal surface around there of the cylinder, or they will be collected on the static particle collector plate, which is on the right-hand side, right before the clean air. And this state of the particulate matter is known as Boltzmann equilibrium, which is a very commonly not known process. And one thing to note here too, I think it's important to, as a kind of disclaimer, if you will, is VFA Solutions, the partner that we recently brought on, was formed in 2007. So it was her business was not formed as a reaction to COVID-19. It was long before COVID that clean air was a right, and that she wanted to help protect people in all different environments. So it's a really great solution for cannabis and for plant health as well. And then when it comes to the third-party testing and validation of specifically the ASPRA system, it can pick up particle sizes of 0.3 microns, which is 30 nanometers. That is a very small particle size. So essentially, it's not discriminating on particle size. It picks up anything large and small. And this, if you can see over here on the right, these images, PM 2.5, PM 10, these major red flag particulate matter, according to the EPA, are all far above that micron size, that 30 nanometers, meaning that everything is being destroyed, picked up and destroyed by this electrostatic precipitation and filtration process. And obviously the botrytis, powdery mildew, aspergillus, same thing. We're looking at 10 microns, so that's obviously going to be picked up and destroyed as well. We also, she has done several tests of the system of ASPRA. And if you look down here on the chart, you can see that the removal rate, the removal efficiency was 99%, which is, again, pretty amazing. Destruction of all these different various particle sizes, anything from, like we saw on the earlier slide, dust to viruses to microbials and bioaerosols. She also, we have an ozone test as well. And because this is closed ionization and not open ionization, and because there is that static filter at the end, to be safe, there is a minimal, almost nothing ozone creation. Because I know that's a concern. You see ozone generators as a common odor control system. This is not that, and we have third-party data to prove that there is no ozone created that would be harmful. We also, another thing to note is that during this test, it was a single pass. And a lot of times we're not just doing a single pass with these systems, with our systems. So the fact that it showed that level of efficiency and no ozone created with one pass just goes to show that's a very conservative test. We are also, the system is as ISO 16890 certified. This is a European, the EN 1822, is the European equivalent of ASHRAE 52.2. I'm sure a lot of you are familiar with that certification. We are currently pursuing that for the ASPRA piece of the technology. So more to come. Actually, really quick, I did want to mention, so one of the benefits of having Dr. Alex Guenther, senior scientist, as well as our chief scientific officer, who's not on the call today, Dr. William Basuete, on the BEA staff, is that they have the intuition and the intelligence to understand with other technologies that may be used for odor control or attempt to be used for odor control, such as photocatalytic oxidation, for example. They raised a red flag to us, and they said, with the application, the concentration levels and the humidity levels of these cannabis operations, PCO might not be the proper air cleaner device. It's certainly not meant for odor control. And so that's one thing that we should just be credit right away. PCO is not for odor control. But if you're looking at it to clean your air in an indoor facility or a greenhouse, whatever it may be, they brought that concern to us. And so we actually studied it and we looked into it. We did a test. And in fact, there were aldehydes that were produced. I think 10 of them went in the intake and in a cannabis setting. And I think 7 of those 10 actually doubled in their concentration levels. And those are aldehydes, as you know, are carcinogens, so extremely harmful to human health. And I don't say this to be like a scare tactic, more so I just want to make sure that when it comes to your odor control system that you bring into your facility, and who you partner with, that they have this understanding and that they have third-party tests to actually show in a cannabis application, because PCO is great for airplanes. This is how we breathe clean air, recycled air in an airplane, but not for a cannabis operation. So there's different competing technologies, if you will, that we really try to better understand and educate the industry on as well. The term aspra, just for everyone's edification, is an old Latin word for mountain air. So it is not an acronym, which is maybe a little bit of a misunderstanding sometimes. And I will end because I know Nadia's given me the eyes, and also it's 5 minutes before. All of our systems, including aspra, are all able to be smart. They can communicate to each other. You can turn them on and off. That way, during blackout, you can power up the system, obviously, power it down during ridge vent opening. Also, it allows for cloud-based monitoring and reporting for compliance purposes and to prove or disprove a complaint that comes in with odor, with regards to odor. That is it. I will stop. Thank you.
Thank you, Emily, and thank you, Alex. We have a few minutes. I wanted to see if anyone had any questions for our speakers today. And you can go ahead and unmute yourself if you want to ask or chat. Nobody?
This is Mark Byers. Can I mention something?
Is it a question?
It's a clarification.
Okay.
The Emily is accurate on our cylinders that we have metal, but we have plastic cylinders. They're refillable. So you can refill the plastic cylinders. I just want to be clear that you could get a scrubber with plastic cylinders, not metal.
Yeah.
But the main thing is that they don't go to the landfill. We refill the plastic. We refill and shake and fill them to the proper weight, etc. So just wanted to clarify that.
Thanks, Mark.
Thank you. Yeah.
So we have a question from Bruce in the chat. What criteria for cannabis odor levels would you recommend to local permitting officials to determine odor standard compliance for cannabis facilities? Is there a portable instrument in existence that can measure 321 MBTs?
Not at the levels that it would occur outside, and especially, again, made difficult because of the large amount of terpenes and other compounds in the air. So that's something that's difficult to do economically right now, although we're working on something to do that. So that's going to require some development, and we recognize that it is difficult, right now when we don't have the quantitative capability to do it real time. But what we can do is you can collect sorbent tubes and send those back to the lab to be analyzed, which is a low cost, relatively simple thing to do. So for right now, that's the approach that we recommend, is collecting sorbent tubes. So you put 1 liter or so air through the sorbent tube. The 321 MBT and terpenes and other things will be collected on there, and you send it back to the lab for analysis, and that works.
Are there labs all over the country that are easy to identify and find and send?
Yeah, right now there aren't, because until a year ago we didn't know 321 MBT was even something people should be looking at. Actually, we found it even difficult to find labs who will do monoterpenes in gas phase at ambient levels.
Wow.
We've been looking around and trying to come up with some labs that would do that. But if there's a market for it, then people will do this. But it's certainly not an easy measurement to make, but it can be done, and that's what really right now seems to be the only approach that's going to work.
Well, the best way to create, one of the best ways to create a market for something like that is for these compliance agencies to require it. So,
Yep.
From Troy had a question about rules of thumb. Emily, you had mentioned, for that greenhouse, 225,000 square foot greenhouse, you know, that 3 air changes per hour, was recommended. Is that typical? And do you just have any sort of guidelines for where to put these canisters?
Yeah. Yeah, so that's the minimum, and that was based on that specific size. So again, it's really going to be based on the number of plants and the canopy size and the square footage of your facility. That will go into that cannabis emissions profile, that mass balance calculator. So as far as number of units and number of air exchanges, that will vary. The 3 air exchanges is really bare minimum what we would ever recommend, even in a smaller setting. So yeah, as far as placement, they can be suspended from the ridge vents. I know a lot of our clients in California, that's what they do. We've seen them, you know, in the aisles in greenhouses, but they're not small. The molecular filtration systems overall, they're fairly large. So we definitely recommend suspending them from the ceiling in a really sturdy frame that we actually build ourselves. And they can be, I haven't been in many of them. I was in a couple of facilities actually, and they're usually along the ends. And then we have a duct sock actually that will go from one system, and that way the clean air, if you will, there's holes at different times on the clock, if you will, on the duct sock that stretches across the greenhouse, and that treated air, that scrubbed air, will then be blown across the greenhouse. That way that's getting the air where it needs to go. It's spreading it across the greenhouse with that duct sock. So that's, you know, the placement would kind of depend on the design of your facility. So it's hard to do a rule of thumb. As you guys know, there's very unique settings
Right.
in the cannabis industry. One thing that I do want to notice, because you talk about indoor versus a greenhouse, and indoor, we would not recommend odor control in the cultivation space. We would only do aspera by itself, which for plant health and indoor air quality, stopping the spread of powdery mildew and keeping worker health secure. Now we would do in the hallways, which you mentioned, that is exactly where we would recommend the odor control and molecular filtration. So we'd put that in the hallways, any type of shared space. Corridors, often what you see in these large MSOs, they've just giant hallways, put them in the corridors, and trim rooms. That's a whole other thing. The dust collection, I really didn't get into too much detail, but we have the capacity to do a lot of high concentrations of dust mitigation as well in those trim rooms.
So Troy, you said, so put them at the source versus the building around the rooms, and that's not necessarily what I just heard Emily say. I think that might be nice to do. What we typically recommend is to put any sort of odor mitigation devices where air is going to escape the building, because you probably don't care about odors in your facility. It's the people outside your facility that care. So, you know, putting them at the ridge vents, putting them in doorways, putting them where you might have a lot of infiltration or exfiltration, air moving in and out of the building. That's where we want to try to capture those VOCs before they escape the building, if possible.
Yes. Yes. Thank you, Nadia.
Yeah, you're welcome. All right. Well, that is our time. Thank you so much, Emily and Alex. I really appreciate you putting this together. Thank you everyone for joining us today. And there will be a link to this webinar available for you to listen to later. So if you have any follow-up questions, I'm sure they are both willing to field them if you send them an email.
Yes. Thank you so much, Nadia.
Thank you, Nadia.
Thank you.
Really appreciate everyone's time today.
Thanks, guys.
Bye.
Bye-bye.